Thank you. We're going to start with this view, which is beautiful city of Melbourne, which I have having lunch on Sunday afternoon. A typical day of Melbourne, you will have a UV index of extreme, because of course we're in summer, right? So you have to take care about that. Humidity will get as low as 75%, but also as high as 86%. And the first morning I was here, I was watching the news and on the weather, right? And said, well, today we're going to have a very soggy day. What did I mean with soggy day? I went out and I understood what a soggy day feels like. That's because there's so much relative humidity around. So in order to understand how that means for life, a way to translate that is to evaluate what we call water activity, in which what is available for life. And you can translate that, in this case, the highest value of 86% relative humidity of 0.686 water activity. Free water, the one you have in the bottom here, will have a water activity of 1, that means that it's available to all life on Earth if you need, right? Compare those values to the city where I was born, which is a more abandoned town now, abandoned nitrogen mining town in the middle of the Hyperadic core of Atacama, in which this very same day we're going to get an humidity as low as 17%. 17%. You can imagine how dry is that. I was telling my colleague this morning that, for example, we are used to a huge amount of static electricity, right? The stuff when you start something when something really high. And as a kid, I would go to bed, I would cover my cheek with my cheek and just move my hand and would generate enough light so I could read magazine. It was that dry, right? And so if we go into the real dry places of Atacama, like this valley in the Hyperrecord, we cannot measure any water at all with an instrument. We have brought a number of instruments available to measure relative humidity. At some point during the day, every day, relative humidity drops to 0. I mean, they are not able to measure any water molecule in the atmosphere. The Atacama Desert is located in northern Chile, and there's a number of reasons to explain why the Atacama is so dry. First, all the masses of air that are coming from Atlantic Ocean, when they meet the Andes Mountain, where Argentina and then Peru and then Bolivia are located, all those masses of air, when they deposit the rain at that side of the Andes, looking at the Atlantic Ocean. And when they come out, those same masses of air to the other side, as they have deposited all the rain to this side of the Andes, at the other side, they get very dry, the so-called rain shadow effect, which is also well known in other mountain ranges across the world. Okay, so you have the first factor there. A similar phenomenon happens when you have all the rain that actually you have here. Once those big masses of air deposit all the water in the tropics, they go up into the atmosphere really high and they go down at the exact latitude where the Atacama is located. So you have a constant dry inflow of really dry air coming from high in the atmosphere. And if you add to this that you have the Humboldt current bringing you really cold water from Antarctica, you might imagine that you will have enough evaporation, that enough clouds, in order to bring any clouds into the Atacama, because also we have this small mountain range, which is the coastal range, which is not any influence of clouds as well. So if you add all those factors together, it's really why the Atacama is so dry. So there are places like this small city, this is called Quillagua, and you can see all the greenery because there's a small river that runs through there, that they have not recorded rain since 620. And there was just one single rain event, about 1 millimeter of rain that took place in the 1980s, that we think that actually was a heavy fog event. So from the time of the Spaniards, those people had never seen rain. And actually, in the city that I was born, because I was born in the Hyperrecord, I moved one city closer to the Andes, which we would have some rain because we were close to the Andes. When it rained, our family would take us in a car to look around to see how it rained, because it was so unusual event, right? So also the Atacama is a really old desert, and that's because of the global climatic patterns since the 150 million years away. you have been more or less in the same latitude, so affected by the same global climatic conditions. So it's not only the driest place on Earth, but also the oldest desert on Earth as well. And if you add the other factor that is characteristic of the camera, we're still understanding why, on UV radiation, this scale, the term, well, UV radiation is too high, now use a hat or use a sunscreen, etc., etc. There are places that I was recently mentioned a few 2 weeks ago, in which because you can measure that as a UV energy, but you can translate it into the same scale, you have a scale of 25. That's a lot of radiation. And you are, again, you're going to ask for mercy if you forget to use the sunscreen in those conditions. So you have still desiccation, you can still UV radiation. Also, many of the soils of the camera are really high in salt. So you have highly soils, highly saline soils, which explains why that the camera is a well-analog model of Mars. We have been studying as such since 20 years already, when the first researchers from NASA came to this test in order to look for proper analog model of Mars, which would test the instruments and rovers. Okay? So my focus is trying to understand, as I was introduced, the close relationship between water and life, because all life, as we know, requires cold water. So this is a perfect model to understand how life is able to survive under such conditions. In places where it seems that you will find anything, because we don't see any evidence of water. So I started investigating this desert from the coastal range. We have a little bit more of high relative humidity, and going inland in order to see how life was able to adapt to that condition. So in front of the ocean, you can see there the Pacific Ocean. You have this phenomenon in which, if you see those black bandings, actually those are a collection of different types of microorganisms that are able to survive there because they are using ocean mist as a source of water. You can see they are very thick, they are about this thick, because they have a constant inflow of water coming every day from the ocean mist. Right? There, relative humidity is really high, between 72 and 92%, because the ocean mist, like Florida. But you don't see all the greenery around itself. Only microorganisms that are able to live exposed to the environment because they have enough water for that. In the same area, there is one cave that is very interesting. We found another type of biofilm, a collection of microorganisms. Even there you can see that green color, typically we tell you that you're finding chlorophyll. There's a microalgae that is able to live there between, and the relative humidity inside between 59 and 89. And notice that we already dropped to 59% relative humidity inside the cave compared to 70 something in the other cave, right? I have to mention here, I'm in Chile. We're working in Spain for 6 years. The year before we inspected this cave, in that very same place, the Princess of Spain came to visit. And a year after I arrived to Spain, when he was king, he visited the place we were working with. So it's a strange connection between me and the Spanish royalty, so to speak. It seems like they brought me to work to Spain for yet on a business I don't understand yet. Also in the coastal range, we have this other cave, which actually we see this is a fracture of an ancient fault, like the San Andreas fault that tracked the earth, and you have a cave formed for that. We found a similar phenomenon there, not at the bottom of the cave, but at the entrance of the cave, we found these green things going on top, not of the cave wall, but on the top of the spiked web that covered the cave wall. When you find something that is green, we suspect that it's maybe chlorophyll. Look at how relative humidity dropped down to 14% during the day, just a few couple of maybe 100 yards going into the desert. Already, you have a big drop in relative humidity available. We think that is happening there because here, the main source of water for lives are the low clouds that are able to go into the hills and then being stopped there. So I already told you, we have something that is doing thoroughly photosynthesis because you can see the chlorophyll on top of the spider web. Have you ever seen a spider web early in the morning? How it looks? It's covered by dew. So this, which is actually a microalgae, learned that in order to survive in the course of the dry desert on Earth, a good place to live wasn't always spider web, because the spider web highly microscopic. That's why it looked like that. Right? It's a beautiful example of how life finds a way, even in such extreme environment, by trial and error probably. And the intriguing thing is that this species of microalgae we found there is the only one of other 25 species that are all live in aquatic environment. This one was able to live in the aquatic environment, live on top of the spider web, because there you will find a source of water. If we move a little bit inland in the same coastal range of the Atacama, we will find a few places in which we will find some veins of quartz stones. Again, the main source of water here is those clouds. Quartz stones are translucent to light, because pick a quartz and you see the sun coming through light, so you can perform photosynthesis under there, help with the fog water that forms around the earth, so you have an entire a collection of species being able to live under the rocks, because the conditions here are now dropping to 60%, 16% relative humidity. Now you have to find a place in order to hide all the still conditions you have under. It's like a miniature greenhouse. The way nature invented greenhouse. And the quartzstone, because you have enough light for photosynthesis, quartzstone, by the way, remain during the day much colder than the environment, so they trap preferentially water from the environment, from these clouds that they will have. So you have light, you have water, you have CO2 from the atmosphere, that's everything you need for photosynthesis. Right? And under any of these quartzstones, you will find at least 70 different types of microorganisms of every type, imaginable type. So you have really islands of diversity at the end of these quartzes, which is also telling how interesting that in this such extreme environment, given the proper conditions, you will find a subspecies there that if you move outside of the quartz vein and you will find no quartzstone, maybe the diversity drops really low. Which is probably the same case we'll have on Mars. Maybe you have particular hot spots in Mars where you'll be able to find things in life, you just move a few feet away and you will find nothing. That's what is trying to find this autonomy. We have been able to grow these type of microorganisms there. You can see again, different morphology, different sizes, telling you the ones that are able to do photosynthesis. From this, we have been able to work with a particular species, which is a type of cyanobacteria that is able to do photosynthesis in order to survive, and we're inspecting that as a model to understand how they're able to survive under such conditions. In the near area as well, you will find this white spot. These are gypsum crusts. Gypsum again is able to trap water from the atmosphere. So you take those gypsum crusts, this big maybe, you crack it open, you will get to see this green banding, again, green chlorophyll, and then you spec out the microscope, and then again, cyanobacteria able to live close enough to the surface to get enough light, but not too close to the surface to get killed by UV radiation, for example, and then be able to trap water from the atmosphere that is condensed in the gypsum stone. Here, now water acidity, relative humidity drops to 1 during the day, and 96 during the night. But this guy found the proper place inside a gypsum rock, close enough to the surface in order to survive inside there. Now, if we move to the real high parallel core of the Atacama, which is this one, here's where we measured 0% relative humidity in every afternoon. And I have a few colleagues that have gotten sick there because they didn't follow my advice, drink water even if they don't feel like it. Because you're losing so much water that you're getting dehydrated really quickly without noticing. And I was joking with my colleagues that in those conditions you can drink gallons of water a day and you will not feel the need to go to a bathroom because no water reaches your kidneys, they're all being evaporated. If you could have a camera in order to see water molecules, you will be like torches of water, losing all the water to the atmosphere that is demanding more than water. Now, this is the site that NASA in 2003 proposed that was a proper model of Mars. I know because I was, in my previous life, I was a winemaker. And I got interested in science when I was middle aged. I turned a hobby into a passion, and that passion into an academic career. So these guys, led by NASA, came to this site and replicated some of the experiments that the Viking mission did on Mars, which is the only mission that went with a specific end of trying to find evidence of life directly in the samples they were analyzing. So they repeated some of the experiments that the Viking did there and found more or less the same result. And because they still have some of the every mission that flies any instrument, of course you have the flight version of the instrument, but you have your backup instrument of your lab, not to decide what the flight instrument is doing up there, right? So those instruments of the Viking mission were still around and functioning. So they wrote some of these on the camera, and they published that the camera was a proper analog model of Mars, because when they replicated the Viking experiment, they found more or less the same results. And that they have found the potential dry limit for life on Earth. I mean, that place so dry that nothing was able to survive it. That second part, I knew that was not true. Why? Because I was born and raised in the country. So I remember that as a kid, we used to travel by my parents because we were going to take our vacation 1,600 kilometers to the central part of Chile, which is Santiago, then to the south, which would be 6,000 kilometers, which would be the equivalent of going to Atlanta for vacations. So we would drive to that region, and we would see this as a common phenomenon, a heavy fog in the region. So I knew it wasn't as dry as they were saying it was supposed to be. And actually, one colleague later found that in that same region, you will find these rocks, halite, which is 99% sodium chloride that common table salt use for your salad in your kitchen. And if that colleague inspected those halite crystals, and inside them, they found another type of material able to do photosynthesis inside the salt crystal. Have you ever left a lump of salt on the top of your kitchen in the morning and see that it turns into like a syrupy sink? That's because salt is able to trap water for the environment. So these guys learned that in order to survive in one of the driest regions on Earth, a good way, amazing, to survive was getting into a salt crystal, because at some point, every now and then, relative humidity in the area will be high enough, so the salt crystal will start trapping water from the environment. So you have a really bright solution in there, in which they could use in order to survive, and for photosynthesis in this case, which was an amazing finding, but no one would expect that. So it took me a few years and some funding, of course, to go to those places, which as a kid I remember never seeing any clouds or fogs, and also places that were also very windy. You can go running in a windy day, your clothes will never get wet because the wind will take all your transpiration away, right? Same phenomenon here. So it's a places with no cloud, no fog, very windy. So I came up with zero that were much drier than the one that described by NASA. This would be the driest. Again, zero during the day, but now we're now ranging the 50s of relative humidity. The way life works is very interesting because this place is just 15 minutes drive from the place where I was born. So it's a full turnaround. Me going to winemaking, then going to science, then going to Gamma, what, and we'll discover the driest place on Earth just in my own backyard, so to speak. So once we knew that was a really dry place, we went into the soil because, of course, we wouldn't expect microorganisms, if there were any in the area floating just around. We dug a pit there in the most Breaking Bad style. And that's not because we are protecting ourselves from something that is on the side. We were protecting the site from ourselves because we knew that we were close to a place that was so dry that maybe nothing was able to survive there. So we wouldn't want to contaminate with our own microorganisms that were on top of us, on our clothes, or instruments. We were very careful of that. So we put sensors on depths. So you will see the typical behavior in any part of the world. You will have relative humidity high during the night, it will drop during the day, and so on. But in this case, up to only 58%. We were having with the sensor that were running 5 centimeters down, or like this, 20 centimeters down, around like this, 50 centimeters down, around like this, and 80 centimeters down, around like this. There, you have a and we performed this experiment for 2 years. Relative humidity was as low as 7%, constant in time, ever the same. And actually, we found there a place was as dry as Mars. So I was saying, well, maybe in this case, we did find a dry limit for life on Earth. The place was so dry that nothing was able to survive it. But we are surprised when we start sampling that site and searching for microorganisms, and again, taking all the precautions to know that actually what we were finding was coming from the south, and not from my colleagues, right? We found a number of microorganisms that were able to survive there, which begs the question, how they're able to do that? I mean, again, look at this environment. Where are they getting their water to survive? And we're trying to understand the molecular mechanisms involved in that, because it will give you an idea of how life may survive on Mars, but also the potential application. Because imagine we can transfer the property to plants. So instead of being irrigated every day, you can irrigate it every month. Once you learn the genetics involved in such extreme tolerance to desiccation, right? So the desert itself showed me how life was able to survive in such extreme environment. And I like this video in particular because it looks like a panoramic picture of just moving from side to side. No, actually this is a video to see that there's nothing that moves around in these areas. Here you can see that there is a video, not just a picture, a picture that moves from one place to the speed of another. So we were driving in these areas, and we went on the street to Maria Elena, which is the right place we could find around. In 2015, we noticed these small lagoons, and since there was so much mine activity around, we got mines of copper and lithium and nitrates and etc. We saw there was a waste of water from some of the mine operation around, so we didn't give it much attention to continue going. But in 2017, it happened, the same thing that happened in 2015 that we didn't knew that was happening, but due to climate change, the temperatures of the Pacific were getting abnormally higher. Warmer water, more evaporation, more clouds, and so what happened in 2017, and that huge mass of clouds came for the first time ever recorded in the history of climate in Chile from the Pacific Ocean and went inland and was raining as never before. And we said, well, maybe in these areas, that amount of water will be welcomed like manna from heaven. Oh, finally, we see water around. Let's grow and multiply. But when we inspected those lagoons, we were expecting just to take a sample of water, things moving all around. We couldn't see anything, and we had to filter gallons and gallons and gallons of water in order to see and find just 4 types of bacteria they were able to live in those water. Other way to say that those water were almost sterile. Just 4 types of bacteria. But here, I just did the experiment, and I will show you later. In every per gram of soil there, you have zillions and zillions of bacteria. Here, 4. What happened with the remainder of bacteria that were living there, including those cyanobacteria I showed you inside those Halley crystals? Since NASA discovered in 2003, there's many works describing the microbiology of the region. What we see that happen here is that these guys are so well adapted to the lack of water that when you give them too much water too soon, they just simply don't know how to deal with it and start getting water into until they die. It's like the same concept when you find a castaway. You cannot, after being alone in the Pacific for 1 month, you cannot invite it to the next barbecue here, eat and drink what much you got. No, you can't. You probably you have real hard problem with health. You have to give it some nutrient solution and then and so on in order to get a problem. We found the same thing because we could find the molecular remnants of the other species that were living there before, right? And actually someone here in the US wrote a poem about the deadly rain because it was so little finding that on Earth we will find life forms that when have too much water they will perish. But in this case it makes sense because we are talking about microorganisms that evolve in the dries of all the deserts on Earth, so they have all the necessary time in order to work to be so good in trapping using conserving water that if you give it too much water, they can simply just do not deal anymore how to deal with it. Okay? So for now, I've already told you that the climate is a extremely arid place. It has extreme UV, it has saline soil, and I forgot to mention that also there's a number of highly oxidant species in such soils. And I left one of the most interesting sites for the end because I once saw this picture from Curiosity taken in 2016, and when I saw that picture, I said, I know that place. I have been there. Actually, this is Redstone. This is a place we found in the coastal range of Atacama. We characterized that site. It's not as dry as the one inside, but it still get dry during the day, 10% relative humidity. Here, you can find this sediment formed by sandstone, mudstone, and rubberite such as halite and gypsum, which tell us the history of this site, which is there is the remains of an ancient alluvial fan that formed under arid conditions. Because you find gypsum and halite, you can tell that when you have that river delta there, it was dry enough for gypsum and halite to be still around. And this had water between 160 and 100 million years ago. I mean, at the time the dinosaurs were roaming the rest of the planet, this place probably looked no better than this one. Just a stream of water, maybe a few bacteria able to survive around the stream, and that's it. So we characterize the habitability. One way to do that is try to culture whatever is there. Of course, it's just a process of being lucky. You don't know what exactly the organism would like to eat. So you try to inoculate different type of growth media with sample of soil. If you were lucky, per every 100 plates with different type of media, you will find a single colony. Compare that to the experiment I just did with the soils of camps there. I had to dilute this 3 times in order to see just a few colonies because there's so much around. The other way you can do in order to spread the habitability is try to extract DNA directly from the soil in the hope that whatever is in there, you will be able to extract this DNA. So what you do there is you start with whatever is in the soils, microorganisms, you extract DNA, you will amplify a particular gene that will tell you the identity of that microorganisms. Once you have the sequence, you compare with the databases, and that will tell you what kind of microorganisms you have. Okay, that's the way you do it. And one way to represent this is say, well, you have here 11 samples, you have more or less of this of that type of microorganisms. Every row will be a microorganism. The darker the color, the more you have for any particular sample. So in our case, we found these different type of bacterial groups. Again, the same concept. For example, those last row, very little of everything. The top row, very a lot of everything, right? The main bacterial group there. But it caught our attention that even though we had those sequences, almost 9% were unclassified, meaning that the database, which nowadays have billions of sequences, could not find anything that looked like those sequences. And let me tell you how you explain this. All life as we know it, we can assign a species name in an orderly manner. So, for example, Homo sapiens, right? That sapiens is the species name, the genus name is Homo, and there were a number of Homo species were the only remaining one. For example, one other being Homo neanderthalensis. Actually, I look more than 100 Homo sapiens. I think of. Then you go to the family Hominidae, which include, for example, orangutan, chimpanzees, and gorillas. You go to the order of the primates. You go everything from orangutans to lemurs. Then you go to the class of Mammalia, which includes everything from bat to whales. Then you go to the phylum Chordata, all that we have a spine, so to speak, that goes everything from snakes to lions. Then to the kingdom Animalia, that goes everything from ants to ostrich. And then to the domain Eukarya, because we have 3 main domains of life. The eukaryotes, we have a true nuclei, and other things that do not have a nuclei, which are bacteria and archaea that are very similar but different. So we have these 3 domains that you can classify until you get to the Homo sapiens here. So when we ordered all our sequences in order to know how much information we could get from the domain to the species name, we found that in gray you can see all the sequences that almost 50% of the sequences that didn't give much information. I mean, for example, in the case of domain, the database could only tell us, oh, so you are a eukaryote, but I cannot tell whether you are an ant, an elephant, or a human being. Is that level of uncertainty? So if you add up with the other unclassified microorganisms, we found a number of, we have tons of sequences which the database could tell what they were coming from. So we coined this new term called the dark microbiome, referring to all the microorganisms we know they are there, in which we have a DNA sequence even, but even then, you could not tell what it is exactly. And actually, that is one of those sequences that we don't find any identity. And again, databases will have billions of sequences to be compared with. And if you search for that particular sequence in those databases, you will find that is an unknown and classified. That's the key you will have. How many ants can you get in there? In order to say, I have no idea what this sequence is. Yeah. So, this is a picture of Mars by taken by Curiosity rover. This is a picture of the Atacama Desert. This is how it looks like on my iPhone. The other way to characterize whatever's in there is to inspect the number of biosignatures you can get from that site. That means all the type of molecules produced by life that you know that come from life on Earth. So you can find it somewhere else, they can tell, okay, so we found, for example, whatever in there. I said, well, we know that this molecule comes only from life on Earth. You will find something similar on Mars. It will be a pretty good evidence that there's something that's coming from something that was or is alive on Mars, right? So if we look for such biomolecules in redstone, first we have to start at the total organic carbon. How much organic could we get for those samples was really low, from 0.02% to 0.1%. The garden soil just outside will have at least 10% of organic matter. And notice how many things we were not able to detect. Not detectable. For almost every molecule, for every type of sample, we try to find that biosignature. But I will focus on three of them. Crystalline phytane, which again were just about limited lecture, 0.01% in just three of the samples, and also hopanes. Why are crystalline molecules interesting? Because crystalline phytane are the degradation product of chlorophyll, coming from either cyanobacteria or microalgae. But we could not find any other evidence of cyanobacteria or microalgae in those samples. Not the cells, not anything, no DNA, etc. So we could only find the remnants of chlorophyll. And also we could find hopanes, could come from a big family called the hopanoids, which are typically the remnants of biological membranes. So what is telling me that probably those molecules come from the ancient microorganisms that used to live in the site when there was water around. Either that's why I could find DNA sequence that I could not compare anything with alive today, that could be a possibility, or the equally interesting hypothesis that maybe the microbiology there is so strange that you cannot compare it with anything today. Both, I would say that probably the first scenario is the one that is most likely to happen. I don't see that life is so strange that you cannot identify here. I think that the molecular remnants are from it is so old that you have enough data from nowadays to say, well, this look like this other thing, right? And that's interesting because you would expect to find such ancient biosignature, because don't forget that you have mudstone. Right? Those are stones made by ancient clays compressed, as you can see there. And the nice thing about clays is that they have this laminar structure that between the sheets of clay you can get things inside, like water or biosignature. By the way, that's why clays are so slippery because you have this laminar structure that can what is in between them, and so they go on this level, right? So as they can get water, they can get also by signature. So we think, as I said, the property we are seeing here are the ancient clays that still have molecular remnants of the species that used to live in this site 150 million years ago. Which is, by the way, why we are looking for evidence of life inside clays on Mars. That's why the Perseverance rover was sent to Jezero crater to specifically see for clays in order to see if it was able to find any type of biosignature in such very ancient clays. Now, once we have characterized this site, we say, well, now we know they have a site that is extreme aridity, extreme UV radiation. We have also gypsum and halite, and we have hematite, this mineral that gave all the site a red color. What two instruments that are on Mars, or to be sent on Mars, would be able to see this very same sample we analyze? So, in the case of SAM, which is the instrument that is able to analyze organics in the Curiosity rover, they barely could detect anything, if nothing at all, depending on the sample analyzed. Sometimes they were able to detect this organic or that organic, sometimes a little bit. In most of cases, they was not able to detect anything. And let's not forget that the version of the instrument we used was 10 times more sensitive than the one that's actually on Mars. So probably the version that was on Mars would not have been able to see anything in this site. Also, we tested another instrument which is called the Mars Organic Molecular Analyzer, MOMA, which is going to be on board the Rosalind Franklin rover that the European Space Agency is going to send to Mars. The same result, barely to detect this or that in the sample, which is telling me that probably we're not doing the best job in order to detect evidence to life on Mars, because testing the very same instrument in a site such as Redstone, they really have trouble detecting anything, knowing that we have life there. What we call a problem of sensitivity. So as you may imagine, when I published this, I made a lot of new friends, right? Telling, well, we need to do better. But actually, this is the way science works. Because now I can say, if you have found negative results on Mars, be careful. Remember Redstone. Maybe life isn't there, but your instrument has not the sensitivity enough, because I will bet you that trying to find evidence on Mars will be much harder than trying to find evidence inside we have here. That way to see it, this is a good way to see the importance of analog model of Mars on Earth. Because if you design a new instrument and you're having trouble, whatever you're designing, detecting anything on Redstone, you don't even try to fly it on Mars. Why for if you're having trouble already here, right? That way, also the idea of bringing samples of Mars to Earth makes so much sense, because you will have all the might of all laboratories on Earth available to analyze such samples, right? Once we get them there. But let's not forget either that those samples are surface samples. The ones are more exposed to the environment, more support of all the type of radiation that Mars has that we don't have, all the temperature fluctuations during day and night that in the case of Mars can go easily between 0 and 90 degrees Celsius, so to speak, which is also the same case that the Viking to Mars. They analyzed the precise samples that were the harder to analyze. And I know that well because I have inspected this really dark place in the Atacama, and the first top centimeters of those soil are close to sterility. You have to go into the soil itself in order to find any type of organism life. The top centimeters have all the UV radiation, all the amazing desiccating and nuclear, etc., etc. So as you can imagine, I will make even more friends with the Mars return, NASA Mars return mission, telling, well, even if you bring those samples, those are no samples you will probably not find anything anyway. But at least I came in time with my own mission, the European Space Agency mission, because that will have a drill that will be able to go 1 meter of depth, about this depth. That will get you access to the subsurface where you would expect if there are any evidence of life, at least, right? So see how in the course of nature the gamma-related humidity is not that you can grow exposed to the environment, decide where you're surviving from ocean mist. If you go a little bit inland, you have to find a rock in order to cover yourself because the environment is too harsh. If you go if farther inland, you have to go into rocks, and if you go to the dry side, you have to bury yourself in order to be able to survive the such extreme condition of marine, etc., etc., etc. Or another way to see it, this is an interesting pattern because we know that Mars had water in the past and it became in time drier and colder. So another way to say this, if you're interested in the different part of the evolution of Mars, I will tell you where to exactly go into the camera in order to understand what was going on at probably what was going on at that age, where you were exposed to the environment or where you're on the surface, which also telling me that what I cannot tell you what we're going to find in Mars, but at least I can tell you what to look for. Dig as deep as you can. Maybe you will be able to find something there. And with that, I am thank you for your attention, and please make all the questions you would like to make.
Great images. So what's the altitude or elevation of the Atacama?
That's a good question. Actually, it goes slowly from the coast of it. You have 6,000 meters. How much is in feet or whatever?
9,000.
9,000 then, and then you go down again to 500 meters. The altitude over here, and then you go slowly going up until you get at some point where you get into the foothills, then you go, and then you get again 15,000. Right? So it's not a high desert of this question, it's rather like this, like that. Yes.
So with the arrival of rain to the Atacama due to climate change, is there a concern that we could actually lose this this time capsule in a way, not only as a lab for Mars, but a time capsule of our own evolution?
Of course. And not only that, again, think about the potential application that we will understand from these guys in a world that we know that we're going to have drought after drought after drought, and then increasing in time and in force everywhere around. And we're losing the kind of microorganisms that will teach us how to survive that. We're losing the one environment on Earth that will teach us how to transfer all those properties, for example, to wheat or maize plant in order to use a fraction of the water that we're using nowadays. So not only about using biodiversity, but also the potential application that could be there. That's what we're doing also.
Yeah, if you wanted to share anything aside from the resolution of the instruments, the people who are designing the instruments, like, is there anything that you can tell someone who is interested?
That is a big question, because what we're looking for now is organics. You find organics and say, okay, this is very important. But let's not forget, we just found this big asteroid, Ryu. You may read in the news. Ryu had every type of organic you can think of, almost. Of high amounts. But no one has claimed that you have evidence of life on this asteroid. So, if you find a part of organic on Mars, would that tell you that evidence of life? No. So what are we looking for exactly as evidence for life on Mars? That's the question I'm trying to move in the environment. I think the only way to do it, without knowing what exactly what you're going to find, is two ways. First, give me long things. If I were to find one amino acid, two amino acid, but if I were to find a chain of 20 amino acids, that would be fine. Since it would be nucleic acid, maybe A and T and A, G, and C, A, D, G, C, and A, and T, that would be a different thing. And the other thing that you could think about finding evidence of life, and actually anywhere, is a way to measure complexity. which is a way to think about entropy, the degree of order and or disorder in any system. We are working against entropy is the way I like the physicists, they mix up all concepts. So entropy is a way of measuring order by measuring disorder. So high entropy means a high degree of disorder. Low entropy means a low degree of disorder that is order. We are here because we are always fighting against entropy. We are highly complex phenomenon, right? That's why we need all the energy daily in order to keep this complexity going on. Once we die, all this has to be highly ordered, goes to the environment again in order to be a disordered state as well. So if we're going to find a way to look for differences in complexity, thus difference in entropy in any system, we could say, well, I don't know what it is made of, but I have measured it, if I can do that, measure the complexity, and say, well, this is too much order to let it display just for pure abiotic that there's no way to life means. So it is a hard pressing question that we haven't been able to define because we don't even know how to define life itself. What is a living thing? What's something that reproduces? Or can we say that this germ fire will start reproducing? Right? Things like that. So if we don't able to look, and we were not able to, there is no common definition of life yet. Now we're looking for life elsewhere. So it's not an easy answer. No, by no means. For me, give me complexity or give me really long things, that would be a good way to go. But again, that would not also be easy to answer.
What's the depth of it? Like, let's say if you were to, within reason, so to speak, right? On like, if you were to have an instrument designed to go out on the next rover, right? So, like, let's say like you have a borehole like 6 meters, where you could try to do like profiles with like amino acid like searches within.
Yeah, but not just in a visual, for example, also we like to see about. I mean, for that, for example, looking for amino acid, look for not amino acid, but change the amino acid, which is different. And we need at least different strategy to look for change in amino acid. I don't recall any instrument that is able to make change in whatever. Just the component. You have to see about a new instrument that you okay, we are going to create this window, everything over 6, well, we will get one instrument. Nothing else for that. Or something like that. In the case of Mars, because you have so much radiation on the surface, temperature is not a problem. Mars is a very cold planet. I mean, come on, we store our food in a refrigerator, right? We keep it there. I have all my bags in a minus 80 degrees Celsius for the same reason to keep it on time. So cold is not a problem. Radiation it is. That's why we're thinking of the proper place. As we have seen in the camera, they can try to find whatever is down there.
So you need some process to be able to remove the sedimentation particles and then some sort of filter where you could just catch things based off the length of the wave.
And the other thing is that trying to see again on the type of biochemical measurement of the light is going to shine, but knowing all what we now know about life are now, because those people will say in a moment of science, for example, all the microorganisms and stream environment were still yet to be discovered. So we could incorporate all the knowledge of how life has been able to adapt for everything extreme on Earth. Maybe we can come out with a new instrument and a new experiment able to detect the changes made by life itself. And actually, still nowadays, we are yet unsure what exactly the light we found. Some say no evidence of life, some say what you consider this not maybe we believe find evidence of life. So again, it's a very open point. For me, the easiest way to see evidence of life, not such as moving. There's no way nature has invented anything made of rock that can crawl around. And actually, it may be the case because there are like 40% of all organisms have a way to move around. Flagella and different other structures, really, they are able to move around. So we were able to get that extra matter of the soil and put it in the center of the microphone to see swimming. That would be undeniable evidence of life. But for some reason, we left all the classic microbiology behind us, thinking about this really fancy methods, they like just take the very big microscope, damn it, put it there, add water and see what you see. Because I said, just figure out look at the formula there. You see signals in moving around at the microscopic level. That would be a way as well.
So I have a very basic question. This
There is
is
no such thing as a basic question.
very far removed from this type of, it's not my domain, I'm a mechanical engineer by training. So I'm a little hesitant to ask this question. I was wondering.
Again, sorry, but there is no such a single
I
thing.
take it.
They just was.
Thank you. So I understand that one of the basis for taking that as a reference for what could be similar to Mars is of course a relative humidity.
Minerality, etc.
Etc. I was just wondering how the presence of high levels of oxygen shifts that or impacts that ability to take the desert as a reference point. And if evolution of life, if it happened in Mars, it would be
Well,
anaerobic or
there's not
something.
enough oxygen. Yeah, that's right. But let's not forget that life was born in an oxygen environment. So the presence of oxygen, we have so much metabolism on Earth, different type of metabolism that do not depend at all on oxygen. So that's not a restricted item.
So to piggyback on the oxygen thing, the Martian regolith is supposedly has perchlorates. Does the substrate here have perchlorates?
Yes, when they come up. Strangely, recently we tried to find perchlorates because they know that all the are supposed to have. But it seems we exported it home. When we exported the nitrates, including you guys. In eventually we are also sending perchlorates. And so most of your aquifers here and other places of work are contaminated by perchlorates. And I think that is the case because as I said, we tried to find it, but we couldn't find it anywhere. But there are some bacteria that have been shown to use perchlorate as a source of energy. So again, that's no problem for life. Again, actually, I was commenting to my colleague, we just isolated an organism and we got this part of perchlorate from sigma, just by playing around with start putting perchlorate different media, and we got one bacteria there, which characterizing that was on for the form of perchlorate. So every time, that's interesting, every time we try to put a limit to life, there will be something that says, no, I don't found the opposite. Too acidic, no. I know bacteria live in a pH 0. Too basic, no. I have a bacteria that live in a pH 13. Too hot, no. I have a so on and so on and so on. Again, and that's in the case of Earth. Put an entire planet with its own room and try to imagine what we could find there.
We found it at the bottom of the Marianas Trench, hydration.
For example, who got upset that you have entire ecosystem that are not emitted from the sunlight? For example, in there, one of the most beautiful examples, they found bacteria that are able to survive from infrared radiation, from the glowing lava you could produce light you can see. Bacteria say, okay, let's use that light as a solar energy. Right? More questions?
Dr. Bustos, we are recording, so in a couple of weeks we will have this out on the website for everyone to view. We can have a few more questions. We are at 7:01. I want to be respectful of everyone's time, but you're welcome to stay and talk.
So most of the examples that you show are looking for chlorophyll, many of them are looking for chlorophyll. If you are really looking for life on Mars, what kind of evidences will you be looking at? Like, what would be some signs that you might be?
I would not expect for something for the synthetic because I just told you that you have to down there. But that is a normal, so to speak, soil. The areas that we have on Mars where we have deep water are concreted walls. That it seems that you have a body of water that, given the proper conditions in summer, that some of that body of water is able to come to the crater wall and get going down. And we bet you that the set of rules for that body of water that is there is completely different. Right? Again, everything yet to be discovered. Okay, more questions here.
Have you done experiments with like these bacteria stuff from conditions that aren't like first gravity?
Not yet, but we're trying to do that with Professor Carmen there. Because there are a few ways in order to simulate microgravity there, but they are not exactly as pregnancy into space. You can see all these rotating machines, they are all changing the vector of gravity, nothing else. They're not. I mean, we haven't invented a way to reverse gravity yet. Our way to flying cars already. No, not yet. The best way is to take things to space. And what we're trying to do exactly that. We're working on that.
I think pressure is probably actually a bigger factor because that's going to affect how fast water can evaporate off.
Awesome. Okay, again, thank you very much.
Thank you.